Good afternoon. And last 2 days, I really enjoyed the presentation, and also really enjoyed these flowers. You know, last Friday was the Valentine's Day in the U.S., and I just bought 3 flowers. Yeah, bought one for my wife and two for my two daughters. Just 3 flowers. So here, millions. So really enjoyed the last 2 days, and thank you, Rajiv. And what I want to do next 20 plus minutes to share some, I think it's a successful story. And because it took us 10 years to get out of orphanage, those orphan crop, peanut. So just see these 10 years what we did step by step. First, I'd like to just express my appreciation to my co-authors. And you can see here, you could say Manish and Rajiv here, and also UGA, Ye Chu and Peggy Isaiah Aikens, and also the Clemson University, Tillery, and Florida University, Dr. Wang, and the USDA Hobart, and the China, those 3 collaborators in China, Dr. Zhang, Dr. Wang, and Dr. Liao. And also UG, the plant pathology, Dr. Cobras. So that's 10 years. So we really started in 2003, a group of legume led by common bean people, came together to Santa Fe, talk about genome. So next year, 2004, the US peanut genomics initiative started. Then this year, I really also like to invite everybody to go to the AAGB 2014. That's going to be in November 10th to 14th in Savannah, Georgia. So you can say it's 10 years. So we get out of the orphanage. So we're from the poor to, just like Rajiv mentioned, we're not poor anymore. So that's the outline. I like to say, first, let's just see the challenge we have. The challenge also is opportunity. And second, talk about the germplasm collection. Also can see the wild diploid utilization to see some successful story. And then give you some historical briefs, those important events, and such as markers, mapping population, linkage maps, and QTLs. And Rajiv also briefly mentioned those, each section. The last, but not the least, I'd like to share with you about the International Peanut Genome Consortium and the Peanut Genome Sequencing Project. The challenge and the opportunity. This map, you can see it's a world hunger map. What you can see also, the superposed those numbers, it's a peanut production. So we have hunger, also we have peanuts there. So it's a challenge is how we can use peanuts to feed the hungry world. The hunger and the malnutrition kill more people every year than AIDS, than the malaria, and tuberculosis, and combine all those diseases. And also, the more people die from hunger than in war. And that's important. Yeah. One child dies every 5 seconds. So that's a challenge for us, how to feed those people. And also from this slide, you can see the 3 major production of peanuts. China's #1, in India, the United States. This slide see the peanut nutrition. And you can see here, peanut has protein, 25% about, and about 50% oil. And how we can use those to feed those hungry people. This is a challenge. You can see this table. 73% area produce, only 45% peanut production. You can see here, it's Africa, Southeast Asia, and India. Those three areas, 73% area, only produce 45% peanut. So we have big room to improve the peanut production. This slide is back 2009, we had a conference in Mali, Bamako, Mali. This slide is kind of really challenging me. You can see the aflatoxin, food safety, aflatoxin contamination in Mali. You can see all the samples from marketplace and the groundnut seed and the groundnut paste. You can see every sample contaminated over 100 ppb. US has had a aflatoxin elimination workshop, annual workshop, started 1988 in New Orleans, Louisiana, but unfortunately ended 2007. So this annual workshop including all crops, the peanuts, corn, cotton, and the tree nuts. But since 2007, each commodity still continue to have their individual workshop. Like next Tuesday, the corn commodity going to have a workshop, aflatoxin workshop next Tuesday. So the challenge also for peanut is the complexity of the peanut genome size. Just like Rajiv showed that the peanut has almost 3G, like human genome. Second one is a allotetraploid, polyploidy. Peanut has the B subgenome and the A subgenome, those two diploid ancestors. So next, I want just to talk about a little bit about the germplasm collection and the wild diploid peanut utilization. Because for the disease, a lot of treats that came from wild diploid. I'd like to use this paper led by Manish Pandey, published on Biotechnology Advances in 2012. This paper, table 1, listed the major peanut germplasm collection. You can see here, it's for cultivated germplasm peanut collection. EQRSAT has almost 15,000 collections. The next one is a wild peanut collection. So EQRSAT has about 500. The United States has 600 here. And the next one is Brazil, has over 1,200. And the next one is Texas A&M, has about 1,200. So how to use those rich resources? Those germplasm really we haven't used too much. How to use those? This just gives you some successful story about the disease-resistant traits from wild diploid peanuts. You can see in 1993, Dr. Simpson released Texas Ag 6 and Texas Ag 7, because those have really good nematode resistance. But the problem is the linkage drag has really low yield and low quality. And until 2001, they released the first cultivar, Cohen, has really good nematode resistance. And 2003 released a nematode. And 2008, Tifgard, released by Corey Holbrook in Georgia. And also another one recently released, North Carolina and Bailey, also from the wild species, has really good multiple disease resistance. like a leaf spot, and white mold. And also, really good story we just heard by Rajiv Washley talk about the GPD-BD4 has the rust resistance and the late leaf spot resistance. And also 2 lines from China, Yuanzhang 102 and 9307. And those again, the Spanish type are from wild diploid collection. So all those really successful, but it's so hard to use this. Why we cannot use it? Because we do not have that molecular tool, to help those breeders to use those wild species. That's another good example. Marker assisted selection, published by Chu in 2011. This successful story to transfer the marker, Howe Lake marker, into the tip guard. It's nematode resistance. So combine the Howe Lake and the nematode resistance. Took about 2.5 years. So you see the marker assisted selection really can speed up the trait transfer from over 10 years and down to about 2.5 years by the back cross method. Because this marker, you can check the heterozygotes, can use the heterozygotes as a donor. So really speed up this transfer. So again, Rajiv mentioned his marker assisted backcross and transfer the rust resistance. It's another successful story. And next, I'd like to spend a few minutes to just go back to see the historical brief, how peanut get out of the orphanage, from the poor to rich. So again, we had the first U.S. peanut genomics initiative in Atlanta. So developed the first 5 years strategic plan. And then 2006, we had the first international peanut conference in China. So expand the effort to international level. And we had then next year, 2007, we had a second year AAGB at advanced in Iraqis through genomics and biotechnology, AAGB. The second AAGB in Atlanta, we developed the international strategic plan, 5 years. Then the next year, we had the third one in India, Ukraine, and then Mali. Then 2010, that's another important historical event, Peanut Genome Project 2010 inaugural workshop meeting in Atlanta. So this meeting, we decided to sequence genome, peanut whole genome sequence. The next year, 2011, we had the 5th AAGB in Brazil. Then 2012, we launched peanut genome sequencing project. Then this year, again, we're going to have this meeting to probably going to complete the genome sequence in November 10 to 14 in Savannah, Georgia. So you all invited to go to this meeting. I just will review, this paper, the peanut express sequence tag, that's 2004. And the historic 2004 Atlanta Genomics Workshop named EST as a priority because that time we didn't have a marker for peanut, no marker at all. So then you can see the August 2011, we had over 250,000 ESTs. The next event, again, this paper report at 2010, the Genome Project launched in Atlanta. So this meeting decided to sequence peanut whole genome. Just to give back to 2014 years ago, the total, the EST 22 million. But you see the peanut, it's all at 1,300. released really just by my group that time. And also, you search peanut 10 years ago, it's total we had 1,700 nucleotides. That's all we had. Then you back to 2011. 2011, so we had 283,000 nucleotides. You search the peanut. So we made a long way. I like to use this paper, just going to be published in Planet Science. And you can see the total SSR markers today, up to date, we have over 1,500 SSR. We still talk about SSR. So we're going to have a SNP and the others. What you can see here is that all the published references just for the SSR markers. And before 2004, we had two papers, 1999 and 2002. The few markers, ESC markers, SSR markers. And for this table, list all the published genetic map to date in Iraqi species. And of course, from here you can see they have the A map and the B map and the AABB cultivated peanut. And from here, all the published references. And we had 3 publications before 2004. One's in 1993, one's 1995, and another one 2001. Really before 2004, we had really poor, had almost nothing. Again, Rajiv used this map, put this reference consensus map, put all 11 populations. And we had 897 markers for this consensus map. And again, this consensus map plus the wild diploid map plus another AABB map. So put all those together. So we had this high density reference consensus map. Also, Raju mentioned this. We had 3,600 markers on this consensus map. Again, this from Rajiv's group, they put the consensus QTL together for drought tolerance, and there are a lot of markers. But again, we have some QTLs, and so how to use those is another question. This one, consensus map, Rajiv also mentioned this one for foliar disease, and his group found 2 QTLs. Particularly, this QTLs is for rust and the late leaf spot. They are using these markers for marker system selection. Next, I kind of like to use a few minutes to talk about my program. So my program covers 2 crops, peanut and the corn, and also the disease and aflatoxins and TSWV, the virus, and also leaf spot, late and early leaf spot. For last almost 10 years, we focused on develop those 2 populations. It's Sound Lake 97R and the NC 94022. You can see the phenotype. And also another second population, tip runner and GTC 20. We call the S population and the T population. So those 2 populations, developed to the real population. And along this line, we had a F2 map published. Use 96 T population. And another map we published use the F5 generation. Also that's use a part of this whole population. This whole population we have, S population we have 352 reels, and the T population we have 248 reels. These 2 populations also used by the genome sequencing project. Those are 4 perennial lines sequenced, and also 143 reels and 118 reels sequenced by BGI. Just showing some traits. And for those 2 populations, you can see it's excellent genetic research material for Hell Lake. And not just for Hell Lake, also for other fatty acids. And some like, palmaric acid. And also it's good for aflatoxin resistance because GTC20 is a resistant line. And you can see this, George Green, for example, 7 days after inoculation, 7 days and 14 days, and after 7 days, it's already colonized by the fungus. But in contrast, you see the GTC20, 7 days, 14 days, 21 days. After 7 days, you can see more or less fungal colonization here, and it's even after 2 weeks. So this line has some resistance to Aspergillus infection. So we have a PhD student. He's going to focus on aflatoxin resistance. He just asked a question, why does Aspergillus flavus in fact produce aflatoxin? So he tried to answer that question. Recently, he published one paper on the frontiers in microbiology. And in addition to that, also we try to study the pre-harvest sprouting. This population, T population, also good to study the pre-harvest sprouting. Also, this is contributing to aflatoxin contamination. You can see here all the germinating before harvest, before dig. But this line, some lines, it's not has dormancy. It's not sprouting. See here again. Also, this line, these two populations is good for TSW resistance. You can see the NC94022, the best resistant to TSWV, and Tifron has a moderate resistance, and other two perennial lines are really the susceptible lines. And also for the early leaf spot, T population, you can see Tifron has some resistance to early leaf spot, but the S population is not good for early leaf spot. But in contrast, S population NC94022 has some resistance to late leaf spot. So we have the collaborator Dr. Cobras. He's the plant pathologist, make sure the disease readings are accurate as accurate as possible. You can see here, and early leaf spot and the late leaf spot, predominantly late leaf spot here. But for 2013, the early leaf spot is predominant, almost 100% early leaf spot. And in this year, it's not, when you work in the field, it's many different traits. You don't know what's going to happen. But 2013 peanut leafhopper burn, it's almost uniformly infect the field. So, but for this population, and some are segregating for this leafhopper burn. You can see here, it's Dr. Nee, as the entomologist, help us to score. You can see here, a leafhopper burn for those plots, but for these plots, it's almost 100% resistant. This the related papers published related to those two populations. I'd like to just go through one by one, like that's the F2 population. We used the 96 F1, F2 individuals. This had 318 SSR loci. And another papers published on TAG. So those two populations, T population, we genotype 158 F5 real, and we had 239 loci. In the S population, we had 190 F5 reals, we had 172 loci. For those two maps, the important thing is we identified the two QTLs. For T population, we identified one QTL contributing about 13%. Another QTL for the S population contributing about 36%. And the phenotype in Tifton, 2010, 2011, 2012, the 3 years, we phenotyped the TSWV in the leaf spot, and also for the oil quality, and the morphological traits like descriptors, and also for the yield component. And that's a preliminary analysis. We still continue, still improving those analyses. Another paper we published just last year, we just used the F2 map and the F5 map against those traits. And interestingly, we identified some QTLs for all three: the thrips, the vector for TSWV, the virus, leaf spot, and the TSWV is the QTL control all three those traits. Another QTL here. For the F population, F map, F5 map, we have QTLs control the TSWA in the leaf spot. Also, we have other QTL, important QTLs. You can see it's kind of consistent. And we have another publication try to identify some resistant gene analog. One is resistant gene analog 121, and also you can see other some markers like SW spotted wheel markers, they linked to same linkage group. So we did some preliminary QTL analysis, and for this RGA121 contributing to TSWV, but those two kind of contributing to a leaf spot. And just give you some update genetic map. So so far we still continue genome having those two populations. And for this S population, so far we have over 200 markers, the 10A subgenome and 10B subgenome. And so we mapped this F2 A gene on the A9, and also the F2B on the B9 linkage group. For the T population, we have so far we have 200, almost 300 markers on this 10A linkage group and 10B linkage group. It's the map here. Just to give you an example, 2013, 2014, what we did is last year we genotyped those markers or those two populations. We phenotyped how many total 3,600 10-foot plots. So we scored a US descriptor, we leaf spotted twice, the TSWA and the leaf hopper, and also yield trait. And also we're planning to do some increase for the international phenotyping in China and Africa and India. How about 2 minutes? I'd like to just quickly go through this peanut genome sequencing project. So there's some collaborators here. Really totally that's over 135 members in 20 countries at 79 institutions. So that's the organization structure. In International Peanut Genome Initiative, we have the 5 goals. And under there, the PGC, Peanut Genome Consortium, we have this peanut genome project. We have 6 components. So that's the executive committee, and led by this 3 chairperson. Dr. Scott Jackson is here, and he's leading this executive committee. So also the Rajiv used these slides in the peanut genome component 1. We have phase 1 and phase 2. Basically, the phase 1 we finished, including the tetraploid and a diploid. And we are testing in the back. The BGI is testing in the back, then move on to the phase 2 to complete this sequencing project. And that's so we done the real 261 reels and also the 4 parental lines, and testing this back by back. And the component 2 by UC Davis as the A and the B genome plus the population is complete. And the combined BGI diploid data, we are right now in the manuscript in preparation. And in there also we're doing the GWAS study, the clone collection of equals at USDA and China. Component 3 doing the transcriptome. Component 4 also for some new technology, sequencing technology, conducted by UC Davis. Component 5, we learned the last 2 days, the phenotyping is the key. And component 6 is the bottom line to develop breeders, toolbox. So you can see it's come a long way, but the phenotyping, beyond the base pairs, the phenotyping. So again, I'd like to invite you to go to this meeting, AAGB 2014. It's going to be in Savannah, Georgia, Marriott. And it's going to be, nice riverfront and really beautiful. I'd like to use this last slide to thank the financial support, primarily these 3 groups: Georgia Peanut Commission, American Peanut Sheller, and the American Peanut Product Manufacturing. The Georgia Peanut Commission really represented the peanut grower. Those 3 segments each contributed $2 million for 5 years. Thank you.